Hello everyone, my name is Nicole Nachif. I am going to give you some immunology background, like a brief summary of the concept I think maybe will be useful for you before the vaccine course. So we are going to speak about some definitions and the structure of the immune system. Then I will describe the innate immune response, the adaptive immune response, and also I'm going to say some general ideas about vaccines. So first of all, about definitions and the structure of the immune system. First, we need to describe which is the main function and the main role of this system. The main function of the immunologic system is to protect the individual from infections, but primarily from microorganisms, not only against microorganisms, but also against known infection substances that are capable of triggering an immune response. So the role is to protect, to defend against microbes and tumor cells too, and to provide homeostasis. We call immunitary response to a response against those microbes, but also to a response against any component of those microbes as macromolecules, proteins, polysaccharides, or any chemical substance that are recognized as a foreign. Okay, so secondly, we are going to make some definitions, what we call an immunogen. So an immunogen is any substance that can elicit an immune response. So any substance that can elicit an immune response is going to be an immunogen or is going to be immunogenic. Then we are going to call antigen to any substance or compound that is recognized by effectors elements of the immune system. It's any substance that can bound specifically to antibodies or to receptors of the cells of the immune system. There is a clear operational distinction between an immunogen and an antigen. Immunogens are substances that can elicit an adaptive immune response, whereas an antigen is defined as any substance that can bind to a specific antibody. Therefore, we can say that not all antigens are immunogenic, but antigens can be immunogenic. An adjuvant is any substance that can enhance the immunogenicity of a substance mixed with it. So these adjuvants can be biological components of the microorganism or can be chemical compounds. Especially some chemical compounds are going to be added to vaccines to enhance this immunogenicity of the antigen. So about the structure of the immune system. The immune system is made up by primary and secondary organs, anatomic barriers, cells, and various molecules. Regarding the organs, lymphoid organs can be divided broadly into two categories, primary lymphoid organs and secondary or peripheral lymphoid organs. Here in the picture, we can see the central lymphoid organs are the bone marrow and the thymus. The bone marrow is localized inside long bones as the femur, and the thymus is localized in the mediastinum, in the middle upper chest behind the sternum. I hope you can see my mouse. Here in yellow is the thymus, and here inside the femur, for example, is the bone marrow. Then we have the secondary or the peripheral lymphoid organs that comprise the lymphatic nodes, which are going to be distributed along the body, the spleen, which is localized in the left lumbar region of the abdomen, here in blue. And also we have something that we call MALT, M-A-L-T, which is mucose-associated lymphoid tissue, which is a diffuse system of a small concentration of lymphoid tissue found in various submucosal membrane sites of the body, such as the gastrointestinal tract, respiratory system, and more. Then we have anatomic barriers that they are from different types, physic, like the skin or secretions as mucus, chemical, that are gastric fluids or proteins that have some activity, for example, bacteriostatic proteins, And also we have biological barriers as the microbiome that lives in our body, for example, in the gut. Then we have the cells that are called leukocytes, and they can be divided into two: granulocytes, neutrophils, eosinophils, and basophils, and agranulocytes, macrophages, mastocytes, and dendritic cells. But also we have the most important group of the agranulocytes, the lymphocytes. Lymphocytes type B and lymphocytes type T. Regarding molecules, we have the antibodies, the system of the complement, and interleukins and interferons. Okay, regarding the innate immune response, the immune response is going to occur when the pathogen can overcome the first barrier of the immune system, the anatomical barriers, which we described in the past slide, in the last slide. We can see here in the next slide how it's going to be like the steps a pathogen has to overcome in order to provoke an actual disease, or the steps that the body is going to trigger to counterpart the action of the pathogen. So first, the pathogen has to overcome anatomic barriers. Then the innate immune response is going to be active. Also is going to be active the adaptive immune response if this the innate immune response is not enough. And afterwards, the adaptive immune response is going to trigger the later stage of the adaptive immune response to actually develop all the actions the immune system can have against the pathogen. So the innate immune response is a type of immune response that is the first one inactivate after the microbe has overcome the anatomical barriers, as I said previously. This infection is going to start when the pathogen breaches the host anatomical barriers, and the innate immune mechanisms that are going to act, they're already there. They don't need to be prepared, or they don't need to be differentiated or something. They are already there, and the pathogen is going to encounter them as soon as it reaches the barriers. So this pathogen is going to be immediately recognized by resident phagocytic cells, and the cells that are going to participate here is the cells that we mentioned earlier, the agranulocytes, neutrophils, eosinophils, macrophages, dendritic cells, etc. This innate immune response, however, do not generate long-term protective immunological memory, but it's accompanied with inflammation. The inflammation we are going to describe in the next slide, but helps to recruit all the effectors of the innate immune response. The cellular responses of this innate immunity act over several days, and during this time, the adaptive immune response may also begin if some antigens of these microorganisms are going to be delivered to lymphatic nodes. In the lymphatic nodes, these cells of the adaptive immune response are going to reside. So when the antigens come to this place, this adaptive immune response is going to be triggered. While an innate immune response may eliminate some infections, an adaptive immune response is going to target particular strains and variants of the pathogens and protect the host against reinfections by using the immunological memory. So the counterpart of this innate immune response is that it doesn't act specifically against a type of microbe. It acts broadly, generally, doesn't distinguish between microorganisms that are similar to each other. It acts against general structures of these microorganisms. The inflammatory response or inflammation is needed for the innate immune response. Inflammatory response are characterized by pain, redness, heat, and swelling at the site of the infection. These four actions reflect the type of changes that the blood vessels in the site of infections are going through. The idea is to increase the blood flow to recruit all the effectors of the innate immune system. It's like the parasite or the bacteria, the virus, Whatever pathogen is there in the site of the infection is going to trigger innate immune response, and the inflammation is going to be the action that call all the effectors to come to the site. The body is going to recruit all the effectors to the site of the infection because it increased the local flow, so the cells, the effectors are more prone to come to the site of the infections, and also it promotes the binding of the leukocytes to the site of the infections to be able to extravasate from the blood flow, from the bloodstream to the site of the infection. When this innate immune response is not enough and the pathogen is still activating the immune system, adaptive immune response also begins. So what is adaptive or acquired immune response? The adaptive or immune response or acquired immune response adapts to the infection's microorganism. This type of response is going to be activated when the mechanism of the innate response have failed. This response, as its name says, adapts to the infectious microorganism, has the ability of enhancing the response with consecutive exposures to the pathogen. It can distinguish between similar related microorganisms, and it generates memory, which is really, really important. It has three main stages, which are recognition, activation, and effector function. It delays timing appear, almost 3 to 7 days. But the property that this type of immune response has is that acts specifically against the type of microbe or against the pathogen that is infected in the moment. So we can divide this adaptive or acquired immune response into two categories, the humoral response and the cellular immunity. In the first one, we have the antibodies and all the secretions secreted by the B cells, the B lymphocytes. These cells are going to produce antibodies as a response against the immunogen. The antibodies are specifically directed against the microbial antigen. The lymphocytes recognize specific antigens. With the help of other cells, they proliferate and differentiate into antibody-secreting plasma cells. So the B lymphocytes, which are like waiting until the pathogen comes, they are going to proliferate and differentiate into antibody-secreting plasma cells. But some of them, instead of secreting antibodies, they are going to remain as a memory cell. Okay, so the antibodies are going to bind specifically to antigens on the microbe. These antibodies are going to trigger the destruction of the pathogen via a specific mechanism. Some B lymphocytes are going to be secreting the antibodies, and some B lymphocytes are going to remain as memory cells, as I told you. The second type of response of the adaptive or acquired immune response is called cellular immunity. The second type is performed by T lymphocytes. T lymphocytes are in charge of controlling the infections or the pathogens that can live within the cells. So these cells are going to control all the cells in the body somehow, in some way, to be able to know if the cells is infected with the microorganism, as a virus, for example, or if the cell has become a tumor cell. So the T cells are in charge of the cell-mediated immunity. Regarding the adaptive immune, but the humoral response, antibodies are the protagonists of this type of response. Antibodies are Y-shaped molecules with two identical antigen binding sites and one constant site, the Fc or fragment crystallizable region. We can see here in the picture, I hope you can see my cursor, the antibody is going to be, the antibody molecule is composed of two identical heavy chains, here in green, and two identical light chains, here in yellow. The variable version of the heavy chain and the light chain, which is here at the top of the antibody, is going to bind specifically against an antigen. And the constant region here in green is going to determine the effector function of the antibody. So we have 5 different types of immunoglobulins, of antibodies, that they are known as immunoglobulin M, IgM, immunoglobulin D, IgD, immunoglobulin G, IgG, immunoglobulin A, IgA, immunoglobulin E, IgE. The function of these antibodies is going to be determined, as I told you, by the constant region. Why? Because if we can think on the structure of the antibody, the variable region, for example, imagine that this is the antibody. The variable region is going to bind to the pathogen, and the constant region is going to remain outside, like facing the other cells of the body. So depending on this structure of the constant region, different cells of the immune system are going to bind to the antibody. So the type of cell that is going to bind to the constant region is the one that is going to determine the effector function against the pathogen. It's not the same that the binding cell is an eosinophile than it's a neutrophile. Okay. So the antibody molecule has 2 separate functions, as I told you. One is to bind specifically the pathogen, and the other one is to recruit other cells and molecules to destroy the pathogen once the antibody has bound. Okay? Here we can see the general functions of the antibodies. So the first and the most direct way in which antibodies can protect against pathogens or the product is by binding them and blocking their access to cells that might be infected by them or simply destroy the cells. So this is called neutralization, and it's a really important protection, for example, against viruses. So the antibody is going to bind to the virus, and this action is going to block the access of the virus to bind another cell and to infect other cells. For bacteria, however, this binding of the antibodies to the bacteria is not enough to stop the replication. In this case, the function of the antibody, all the antibodies that are going to bind, here we can see in the picture, all the antibodies that are going to bind to the bacteria, is to do like a tag of them. So when the effectors of the immune system see all the antibodies binded to the bacteria, they are going to see, okay, this concentration, all of these concentrations are labeling the bacteria for destruction. This action is called opsonization. The third function that antibodies do is similar to opsonization in the way that they bind to the bacteria and they label the bacteria, but instead of recruiting other cells, this constant region of the antibodies that are outside the bacteria are going to activate another proteins and other molecules that are called the system of the complement. And these molecules are going to trigger the destruction of the bacteria, as we can see here in the picture. Here you have like a little chart with some activities or functions of each antibody. But the most important thing we have to remember is that IgM is the first antibody in being secreted. Later on, we are going to explain why and how it is. But it's the first antigen, the first antibody that is secreted by the B cells. Therefore, when we do a blood test or like antibody titers in the host body, if IgM is there, it's showing to us that the infection is right now. The infection is in an acute stage. And then we have to remember that IgG is an antibody that is secreted later after IgM. So if we have an IgG positive in serum or in blood, it demonstrates that the infection already began or is a past infection because IgG is a memory antibody. Now we are going to explain why. Also, we have to remember that IgM cannot cross the placenta, so a baby is not going to have IgM from its mother, but IgG, yes, can cross the placenta. So it's going to give to the newborn some special protection against pathogens. So the most important fact about the adaptive immune response is that it generates memory. How it is? Because, for example, as I explained how the antibodies, the B cells were producing antibodies, some of them are going to remain as a memory cell, as a backup. The body makes like a backup for later exposures. Imagine that we are describing a first encounter of whatever pathogen you can choose. For example, hepatitis B virus. My body is going to encounter for the first time this virus. The B cells are going to prepare specific antibodies against it. First of all, my B cells are going to secrete IgM antibody. So here in the graphic, in the picture we have here, we are going to see this first stimulus, the virus enters in my body and my body secretes IgM. Also, some B cells are preparing a better antibody, a more specific antibody with different effector function antibody, and that's IgG. So my body, when it's ready to secrete IgG, is going to lower the production of the IgM and is going to produce IgG to my body. When the infections it's already done and my body fought against the virus, IgG is going to diminish because the antigen stimuli is not there anymore because the infection is done. My body could fight against the virus, so the IgG levels remain lower. But they are not going to disappear. Why? Because some B cells remain in my body as a memory cell. If I encounter the virus again, here we can see second stimulus, my body doesn't have to secrete IgM and prepare again to have a better antibody. My body already have a better antibody, the best one, the IgG that they already prepare. So the B cells that were as a memory backup before, now are going to secrete higher amounts of IgG because they already know how to do it and they have enhanced that response against the virus. So here we can see the second titer of IgG after a second exposure is going to be higher and it's going to be only IgG. So immunological memory is perhaps the most important consequence of an adaptive immune response because it enables the immune system to respond more rapidly and effectively to the pathogens that have been encountered previously. And it also prevents them from causing the disease. This is a particularly clear response of B cells because the antibodies that are made during the secondary and subsequent responses exhibit distinctive characteristics such as higher affinity. A primary response is characterized by the initial rapid production of IgM and slightly delayed IgG response, as I explained it before. The secondary antibody response is characterized by a higher production of larger amount of IgG antibody. At the beginning of the secondary response, antibodies are made by memory B cells that were generated in the first response against that pathogen. The secondary response is characterized by a more vigorous and earlier generation of the cells that are going to produce that antibody, thus accounting for the almost immediate abundant production of the IgG. Okay, as a summary, here in the picture, we can see when When it's a first encounter of the pathogen, the body is going to secrete IgM because it's the first antibody that doesn't need to be prepared somehow. It exhibits an immediate specific bounding to the antigen that is stimulating it, but it's not going to be an effector function specifically against that pathogen. That's why the B cells are going to differentiate and moderate and choose the constant region of the antibody that is going to trigger a specific response against that pathogen. After IgM, few days, and IgG is going to be secreted. The response is going to be until the infection is controlled. Once the infection is controlled, B cells are going to remain like in pause. And then if there is a second exposure to the same antigen, these cells that were like in a waiting state are going to immediately secrete IgG again, or the specific antibody that was against that pathogen, creating a really rapid and a really quickly response against the pathogen, avoiding the disease because it controlled the infection specifically and effectively. Regarding vaccine, it's important to say that the deliberate induction of an immune response is known as immunization. We call vaccine when we stimulate the host to generate an active immune response against an antigen. For example, we stimulate a body to generate antibodies against hepatitis B virus. The two most important contributions to public health in the past 100 years were sanitation and vaccination, and have markedly decreased death from infectious disease. However, infectious disease remain the leading cause of death worldwide. Modern immunology itself grew from the success of Edward Jenner and Louis Pasteur because they created vaccines against smallpox and chicken cholera. Smallpox is also called variola. On 1979, the World Health Organization have announced the global eradication of variola because of vaccinations programs. So vaccines are the most important tool we have as health workers to avoid epidemics or pandemics or diseases in general. A successful vaccine must possess several features in addition to its ability to provoke a protective immune response. First, it must be safe. Vaccines must be given to huge numbers of people, and relatively few of whom are likely to die or sometimes even catch the disease that the vaccine is designed to prevent. This means that even a low level of toxicity is unacceptable. Second, the vaccine must be able to produce a protective immunity in a very high proportion of the people to whom it's given. Third, particularly in poorer countries where it's impracticable to give regular boosters of vaccinations because of they are living in rural populations, a successful vaccine must generate long-lived immunological memory. This means that the vaccine must prime both B and T lymphocytes. Fourth, vaccines must be very cheap if they are going to be administered to large populations, in order to be affordable to healthcare systems. Vaccines are of the most cost-effective measures in the healthcare, but this benefit is eroded as the cost per dose rises, logically. Another benefit of an effective vaccination program is the thing that we call herd immunity. Herd immunity confers on the general population. The idea of herd immunity is lowering the number of susceptible members of a population. Therefore, vaccination is going to decrease the natural reservoir of the infected individuals in that population. So it reduces the probability of transmission of this infection. Even unvaccinated members will be protected because their individual chance of encountering the pathogen is going to be decreased. However, the herd immunity effect is only seen at relatively high numbers of vaccination within a population. For example, for mumps, the necessary level is estimated to be around 80%, and below this level, sporadic epidemics can occur. So these are going to be some general points for you to know before vaccine course. I hope this will be useful for you. And please, if you have any doubts or any concern or something, you can contact me. I will be there. And see you. Bye bye.